Okay, so now next we'll be talking about the male reproductive structure in the flower. We already know what the male reproductive structure is, right? Stamen. Okay, so we'll be talking about the stamen now. Now stamens, they are the male reproductive structures, and stamen is also called as microsporophyll. What is the meaning of microsporophyll? अब यहाँ पे देखो, the term fill, what does it mean? It means leaves. Okay, so stamen is the structure where the microspores are going to develop. So what is the term that we are using for the stamen? Stamen is also known as microsporophyll. It is the structure where the development of the microspore is happening. Okay, so stamen is also called as microsporophyll. It consists of 3 important parts. Now let's have a look at the structure. So यहाँ पे देखो, see here, stamen. There is anther, right? And there is filament. What is this filament? Filament is the stalk. Okay, so anther and filament, of course, these are the 2 major structures of the stamen. Now, what we have done is we have taken a section of the anther. So, अगर मैंने anther को यहाँ से cut किया like this, what is going to happen? I am going to see a structure like this. Right? मुझे उसके अंदर की सारी चीजें visible हो जाएंगी. Right? So anther has been sectioned and this is the view that we are going to get on the inside of the anther. So 2 important structures, anther, filament. फिर यहाँ पे एक और structure present होता है which is called as connective. Okay, which is called as connective. Connective is nothing but it is a strand of sterile tissues. उसके अंदर हमें vascular bundles मिलेंगे. देखो, connective क्या है? It is a sterile tissue between the lobes called as anthers and extension of the filament containing the conducting strands, right? We all know what conducting means. We're talking about the conducting tissue. So here in the connective, what are we going to find? We are going to find the conducting strands, and what this connective is nothing but the extension of this filament, right? This filament is getting extended. उसी का extension है, उसको हम connective बोल रहे हैं. And what is it containing? It is containing your conducting strands. Now moving further, filament stalk that bears the anther, right? We can see it in the diagram also. This is the stalk, and what is it doing? It is having the anther. Now, anther, very important. This is very important. Okay, this is very important. Now see, there are certain terms that you have to remember for anther. Anther is bilobed. How did we see the anther as? This is how the anther is, right? This is lobe number 1, this is lobe number 2. So what is the term that we are using for anther? The term that we are using is that it is bilobed. Diathecous structure at the tip of the filament. ये तो हमें पता चल ही गया है where the anther is present. It is present at the tip of the filament. Right? Now another term, diathecous. So what is the meaning of this diathecous? See, theca means covering. Okay? Theca means covering. So here, you know, whenever we are going to see the section, suppose this is the section of the anther, right? In each lobe of the anther, we are going to find these structures, these spherical structures, which are called as the pollen sacs. And यहाँ पे here, what are we going to find? We are going to find the coverings. Okay, so each lobe of the anther is diathecous. Theca. What is the meaning of theca? Theca means covering. Okay, theca means covering. Alright, so anther, bilobe, diathecous, very important NCERT terms hai. So you have to remember bilobe, diathecous, structure at the tip of the filament. Now each lobes, right? What did I tell you? That anther के पास कितने lobes हैं? 2 lobes हैं. Lobe number 1, lobe number 2. Right? Each lobe has 2 pollen sacs or microsporangia. ये देखो यहाँ पे. This is lobe number 1, this is lobe number 2, right? We can clearly see in lobe number 1 and lobe number 2 these sac-like structures. How many? In each lobe there are 2 sacs. So that is what this line is saying. Each lobe has 2 pollen sacs called as microsporangia. What is this? This is microsporangia. Okay, this is microsporangia. Okay, now next, a total of 4 microsporangia are present in the anther, right? हम लोगों ने देखा था section of each lobe. Here we are going to find a connective. Here we are going to find the microsporangias or the pollen sacs. Right? In an anther, how many pollen sacs or microsporangia we are seeing? We are seeing 4 microsporangia. So the term that we can use here for this is tetrasporangiate. Okay, the term that we are going to use here is tetrasporangiate. So total of 4 microsporangia are present in the anther. Each microsporangia produces spores which later develop into pollen grains, right? So the entire story of the development of pollen, कैसे microspores बनेंगे, what is microspore mother cell or pollen mother cell, that we are going to see in the coming up slides. So each microsporangia produces spores which later develop into pollen grains, right? हमारे लिए pollen grains important है because pollen grains are the ones which are carrying the male gamete, right? And for the process of fertilization to happen, what do we need? We need the male gamete and the female gamete. All right. So 3 important terms that you have to remember about this stamen. First important term is that it is bilobed. It is bilobed. Second important term is that it is diathecous. Third important term is that it is tetrasporangiate. Okay, it is tetrasporangiate. So these 3 terms are very important for the stamens. Now next we'll be talking about the development of the anther, right? We have seen that the anther, what anther is. Right now we'll be talking about how the development of the anther happens. Okay, so now see, anther appears as outgrowth-like structure in the initial stage which shows spherical or oval-shaped structure. ठीक है, हम लोग बहुत initial stage के बारे में बात कर रहे हैं. And how is the anther appearing? It is simply appearing as a outgrowth. It is a mass of undifferentiated cell and homogeneous meristematic cells. Now, what is the meaning of this? Undifferentiated cells. Cells are not differentiated, right? They do not know how their shape is going to be, what their function is going to be. Homogeneous meristematic cell. We know what is the meaning of meristematic. The cells continue to divide, right? This you have already studied in grade 11th. What are the 2 types? What are the 2 major types of tissues? Meristematic tissue and permanent tissue, right? So the initial part that we are talking about, the initial, the outgrowth-like structure that we are seeing, it is nothing but a mass of undifferentiated and homogeneous meristematic cell which is surrounded by a single cell thick outer layer. And what is this single cell thick outer layer called as? Epidermis. Okay, it is called as epidermis. Now, first of all, vascular tissues are formed in the middle region, right? Vascular tissues are very important. Food and water has to be reached to these structures. So first of all, vascular tissues are formed in the middle region simultaneously. Now the important story starts. Simultaneously, group of cells located just below the epidermis, right? This is my epidermis. These are the cells which are present here. Okay, these are the cells which are present just below the epidermis. What is happening in these cells? This is my epidermis. Now let's see what is happening in these cells. Now simultaneously group of cells located just below epidermis in vertical rows in the region of hypodermis. राइट ये एपिडर्मिस हो गया. एपिडर्मिस के नीचे वाला जो रीजन है उसको हमने हाइपोडर्मिस बोल दिया. एट द 4 कॉर्नर्स they become large, they have a visible nucleus with dense cytoplasm. So what is going to happen here at the four corners, right? ये हमारे सारे चार corners हो गए. At the four corners, one of the cell is going to become very prominent. Okay, it will have a dense cytoplasm and it becomes very large. Okay, now moving further, these cells, they divide periclinally to form the primary parietal cells below the epidermis and primary sporogenous cell towards the center. Right, so what are we talking about? This is the initial part. These are the cells which are present in the hypodermis region. Now what has happened? One of the cell has become prominent, right, in all the four areas like this here. Now I'm talking about this cell. What is going to happen in this cell? There will be a division. Okay, it is going to divide periclinally like this. It is going to divide periclinally and it is going to form two cells. The primary parietal cell, one is the primary parietal cell and the other one is your primary sporogenous cell. So this is primary sporogenous cell. Okay, so what has happened? The cells which became very prominent here in the four corners, in them a periclinal division has taken place और वो दो parts में divide हो गए हैं. What are we calling them? Primary parietal cell and primary sporogenous cell. Now very important, what is the location of each of them? देखो यहाँ पे अगर हम लोग करेंगे, so this one is your parietal cell and this one is your sporogenous cell, right? So now see, we all can see from this particular diagram what is going to be the location. The sporogenous cell, the primary sporogenous cell is towards the center and the parietal cell is below the epidermis. This is my epidermis, right? So parietal cell is coming below the epidermis. So please remember what has happened till now. We got four prominent cells. These cells are called as the archisporian cells and in them division has happened. Periclinal division has happened and it is forming two cells, primary parietal cell and primary sporogenous cell. Now moving further, both of the cells usually undergo further divisions to form complete structure of anther except epidermis. This is a very important point. Except epidermis, we saw what has happened. We got a prominent cell in the four corners, uske andar division hua. We got a primary parietal cell and primary sporogenous cell, right? Now what is going to happen? Further cell divisions are going to happen. They are going to form all the structures of the anther except epidermis, because epidermis was already present, right? हम लोगों ने देखा था cells है, right? There is a meristematic tissue और उसके ऊपर एक outer layer already present है, which is epidermis. So these cells, they are going to divide further to give rise to the other structures of the anther except epidermis. Okay, now see here. Primary parietal cells undergo further periclinal and anticlinal divisions, right? What is periclinal and anticlinal? This is your periclinal division, this is your anticlinal division. So what is happening? The primary parietal cells, now this is important. You have to make note of which cell is dividing to form which structure. ठीक है, तो यहाँ पे primary parietal cell is dividing and it is going to form a series of 3 to 5 layers making the walls of the anther. We'll be talking about the walls of the anther, what is, endothecium, middle layer, tapetum. We'll be talking about all of them. Now moving on to primary sporogenous cells. So now see here, the primary sporogenous cells, they divide twice or more than two by mitotic divisions to form sporogenous cells, and later the sporogenous differentiated into microspore mother cell during the formation of the wall of the pollen sac. So what is happening? What is this story about? बहुत simple है. Sporogenous cell था. It was towards the center, right? What you have to remember, these sporogenous cells, they are going to differentiate into microspore mother cell. Okay, or we can say that they are going to act as microspore mother cells. Okay, so sporogenous cells differentiating or acting as microspore mother cells, and of course what these microspore mother cells are going to give rise to, these microspore mother cells are going to give rise to microspores, which are going to develop into your pollen grains. Okay, so that is what is written here. So what you have to remember is that sporogenous cell से हमें क्या मिल रहा है? Microspore mother cell, and of course as we can see the name mother cell, this microspore mother cell is going to give rise to microspores. Now see next, each microspore mother cell divides to form 4 haploid microspores. The details of this process we will be reading under microsporogenesis. In brief, this is my microspore mother cell, right? I can write the short form microspore mother cell. MMC को हम लोग PMC भी बोल सकते हैं. What is the meaning of PMC? Pollen mother cell. Okay, so what is happening? Microspore mother cell, it will undergo meiosis. ठीक है, meiosis होगा और क्या बनाएगा? 4 haploid microspores बना लेगा. 4 haploid microspores बनाएगा. Okay, so each microspore mother cell divides to form 4 microspore or pollen grain by meiotic division or reduction division, right? Gametes. We are ultimately forming what? Gametes. So they are haploid. Now please remember, if a question comes and it is asking you about the ploidy of MMC, this is diploid. Ploidy of microspores is going to be haploid, right? This is how we represent it, 2n and n. So each microspore mother cell divides to form 4 haploid microspores or pollen grains by meiotic or the reduction division. This is important. Which division is happening here? It's meiosis which is happening here. Okay, so now see here. This is the epidermis. This is the transverse section of a young anther. ठीक है, यहाँ से हम लोगों ने हमारी story start करी थी. Okay, transverse section of young anther. These are all the meristematic cells, right? It is nothing. In the earlier stages, it was nothing but a group or a mass of the meristematic cells surrounded by epidermis. ठीक है, यहाँ पे देखो 4 corners में. Here, what are we finding? We are finding these prominent cells. Okay, what are these cells called as? Archisporial cells. Now what is going to happen in these archisporial cells? Cell division, periclinal division happens, and we get 2 types: primary parietal cells, primary sporogenous cells. Primary sporogenous cell is towards the center, primary parietal cell is towards the epidermis. ठीक है, और साथ में simultaneously what was happening? Vascular tissue was developing. यहाँ पे देखो, right? Differentiated primary parietal and primary sporogenous cell. Okay, so this is about the development of the anther.